Go.
Yep. Go ahead.
All right. So now for something completely different. I'd like to talk about a project that is very close to my heart. Genetic modifiers, I'm going to make the statement, cause chronic epilepsy and haploinsufficiency mouse model of tuberous sclerosis complex. So before I get into it, I would like to say that although I'm giving this talk, I'm giving it on behalf of Montana, who is first author on our upcoming publication on this project. Montana did this project as part of her PhD research, a significant part of her PhD research, and so any details and opprobrium that this project should generate should be directed to her and not to me. We have some funding sources. We also have other friends, but Montana's who you want to talk to. Okay, so with that out of the way, what is tuberous sclerosis complex? Tuberous sclerosis complex is a multi-system disorder. It's a rare genetic disorder, and among many things, it has it's a neurodevelopmental disorder among many other things. And in particular, it causes epilepsy, presumed to be through features, abnormal developmental features called cortical tubers, tubers like potatoes, that develop in the brain and presumably disrupt brain circuits. That's not the only bad thing that happens in TSC. There's, if you happen to survive your childhood with epilepsy, you can go on to develop neoplasms of the kidney, of the lungs, the liver. It's a bad disease. Okay, we all agree, bad disease, let's cure it. Okay, so genetically, what causes TSC? Well, TSC is caused by mutations in, wouldn't you believe it, genes called TSC1 and TSC2. These two genes, what they do is they code for proteins that form a heterodimer complex, and that heterodimer complex suppresses the mTORC1 complex. So as I probably don't need to say, but I will, the mTOR complexes are sort of major regulatory hubs, controlling a huge amount of cellular physiology, if not controlling, at least modulating it, and various kinds of signaling decisions are at least in part inflected by mTOR, and so if you lose appropriate regulation of mTOR, really in any of these upstream genes that are regulators, you can have abnormal brain development and epilepsy, autism, and a huge number of sequelae. In fact, there's a large set of genetic epilepsies that are called mTORopathies because it's one of the largest genetic classes of epilepsy that happen not to have anything to do with channels and receptors. Okay, so we know a lot about the genetics of TSC. Genetically, it's inherited in an autosomal dominant pattern. So I'm lucky, in contrast to Jackie, who has to model recessive disorders. This one's a little bit easier. You have, if you have one bad copy of TSC1 or TSC2, you have a high probability of developing symptoms of TSC. About 2/3 of patients have sporadic mutations. And most of those mutations are in the TSC2 gene. Of course, once you have them, if you have them at the germline, you can pass it on. However, TSC1 is mostly inherited. TSC1 mutations tend to be associated with less severe disease. TSC2 is a more severe disease, but TSC1 is the more commonly inherited version. Okay, but why am I here instead of at some TSC conference? Because you're all much more fun to hang out with. But TSC has a very highly variable clinical presentation. So there's a bunch of stats here. I won't go through all of them. But there's a very high prevalence of these brain malformations. There's a very high penetrance of some form of neuropsychiatric disorder, although even though intellectual disability is very common with this particular disorder, there's a large fraction of patients with TSC that have normal IQ, for example. And, if you look at the current numbers, up to about 90% of patients have epilepsy. It's a rare disease. It's hard to get epidemiology on this. But there's a substantial fraction of patients that actually have these mutations and don't present symptoms, or present very limited symptoms. And so there's something else going on. And those something else's could be any number of things, but obviously we think that genetic modifiers are a significant contributor. And in fact, there is some epidemiological data, twin studies, et cetera, to suggest that. But before we can do any kind of modifier screen, we have to confront one major problem, which is that mouse models of TSC, and I'll say it here, you heard it here first, folks, TSC models are completely inadequate. So only one study has shown spontaneous seizures in heterozygous mice. So if you knock out one copy of this gene, just like humans have one copy of this gene knocked out, TSC1 or TSC2, absolutely no mouse model has had a spontaneous seizure. Yeah, you can induce seizures, and yeah, they happen a little earlier and for a little longer, yada, yada, yada. But the mouse models do not recapitulate that to date. One study has reported spontaneous seizures in a mouse. I've got the PubMed ID here. This was in pre-weaning head-fixed electrophysiology. It's like a super stressful protocol. And every one of the mice that they studied, actually the seizures resolved by the time basically that the mouse could thermoregulate. It seems to be at most an early resolution of epilepsy, which absolutely is not what human patients have. Human patients have chronic epilepsy. All other seizure models, including models that are being used for preclinical testing, have some kind of conditional loss of heterozygosity. So they use a Krelok system, for example, to turn off this, or to cut out the second copy of TSC1 or TSC2 in an astrocyte or a neuron. And sure enough, you can get epilepsy that way. But as Montana can tell you, there is an ongoing controversy about whether or not that's a face valid model of this disorder, because when you actually look at affected brain tissue from patients, it's not at all clear that there's that loss of heterozygosity is correlated with any of these features. And so it's at best speculative that the current seizure models with the loss of heterozygosity, this conditional loss of heterozygosity, do they actually model what we want to model? Of course, I know Catherine Kazarovsky, and I know she's got some, huge breeding train and, huge research program doing the AD-BXD. And so my big idea was, oh man, I'm going to be Catherine Kazarovsky 2.0. I'm going to do TSC-BXD, and we're just gonna, we're going to ride that train. And so, we've built our first little engine. But the basic idea of these modifier screens, for at least for dominant disorders, is that you have some model. If you have a homozygous loss of function in TSC1, that's embryonic lethal. I forgot to mention that. So you have to propagate those as heterozygotes, but you can breed those to any old background you want and inject some genetic diversity in that way. And what you get when you do those matings is you get probe TSC1 knockout, probe by TSC1 knockout mice, F1 mice, and then litter mate controls. So you genotype for TSC1, and then you're good to go. And so we posit that, of course, outcomes are going to vary. Obviously, outcomes are going to vary. Sure, absolutely, they have to. So we did an itsy bitsy strain survey of 3 inbred strains. I can give you chapter and verse on why it was an itsy bitsy strain survey. It was supposed to be a little less bitsy, but we ended up pandemic. That's part of the answer. Not the shameful part of the answer, but it's part of the answer. But basically, the idea was, well, gosh, as many people in this room happen to know, the DBA2J strain is hugely seizure susceptible. By basically any way you can induce seizures in a rodent, the D2s are massively seizure susceptible. And so the idea was, gosh, that's a slam dunk. We'll cross D2s to our B6 TSC1 heterozygous mouse. And sure enough, that mouse is going to have seizures, prove heritability, off to the races, Catherine Kazarovsky 2.0, on to Nobel Prize, right? What could be better? A man with a plan. Well, we threw in another strain because we could get a hold of it. We threw in BXD87. We were going to throw in a few more pandemic, other shameful secrets. Um, and so we threw in BXD87 primarily because we looked at some BXD strains and we looked for outliers for brain anatomy abnormalities, behavioral abnormalities, and sure enough, 87 um, is an outlier for those traits. Interestingly, it's also one of the most susceptible mice in the ADBXD population. So we had at least some indication that, hey, this might be a fragile mouse. Maybe let's throw it in there. Let's see what we do. As it turns out, we lucked out big time. So my original hypothesis, the one that I was buoying Montana's PhD student spirits with, was like, we're obviously going to have seizures when we cross those D2s. Let's just do it. And then, so of course she did, and we didn't get any seizures. And, that kept me up at night. I'm sure Montana slept well, but I was really struggling. And because it's all about me, I had to address that. And so we said, okay, fine, we're kind of running out of money, we're running out of time, we're running out of patience. Let's try this BXD87. We plucked it out of the lineup. Let's give it a try. And sure enough, BXD87 hets, the mice, the cross with the TSC ones, those mice had seizures. It's not completely penetrant. It's not it's interestingly sex specific, which is not modeling the human disorder, but we observed frank behavioral and electrographic seizures in 4 out of the 7 BXD87 het females that we tested. And it wasn't just like we saw a seizure, we saw like 70 seizures in those 4 mice. And so this seems to be a robust but incompletely penetrant and sex specific effect. Okay, so that's, a little on the weird side, but that means there's all sorts of fun genetics to do, which of course is going to be on conclusions in future directions. One thing that I will say is that the seizures are highly behaviorally stereotyped. I don't know if people are used to looking at mice having seizures, but there's a particular sequence of events that's pretty well characterized. And of course, you can, it starts with sort of a hunching back, and then the tail becomes rigid. It's called Straub tail. Then there's something called a loss of righting reflex, where the mouse loses its ability to stay on its 4 limbs, and it sort of falls over. And then there are tonic and clonic kind of limb movements. Eventually, these seizures resolve. The mouse is kind of quiet for a little while, but then it goes about its business. It's not, we didn't observe any terminal seizures, for example, which can happen in induced protocols. These mice got up and walked around, and we performed behavioral experiments with them and so on. I'm not showing the behavior because that was a real goose egg, bummer. But we did happen to do a couple more things. One thing we did was we looked at histology in the cerebral cortex. And so what we're showing here is just an example of our workflow. So we did sections through the somatosensory cortex just to get a look at cortical architecture, stain them with Crescel violet, did a series of Fiji steps, and we tallied up a few things: cortical thickness, cell sizes, and cell densities in the cerebral cortex. And all 3 of those measures differed at least in one strain. In particular, the BXD87s had significantly thicker cerebral cortices, the hets versus the wild types, which has been observed in other mouse models with seizures. But some of the other cytological phenotypes, like cell size and cell density, seem to not be as pronounced in BXD87, but the D2s were there. So there is some heritable variation in how these different backgrounds are responding to the TSC mutation, but not all of it leads to epilepsy. Interestingly, when we asked about these particular phenotypes, do they correlate with the presence or absence of epilepsy? The answer was no. So here's just some examples looking at some of those. I won't go into this. These are just, these pink bars here are, where we actually grabbed ROIs to do these quantifications. We didn't want to do it whole slide wide, because then you'd have to register things and be, really persnickety about it. But here are some examples of the ROIs that we selected. And what you'll notice, for example, is that this female het mouse, this is a representative example, this mouse, the cerebral cortices of the female het mice were higher on average, but that was also true of the male hets in BXD87. But that didn't seem to correlate with the presence or absence of seizures. But if we zoom in and we kind of put on our Mikhail's, squinting goggles, I believe he called them, or in any case, your phrase was very, if not memorable, it made an impression. Obviously not memorable because I can't remember it. But if we put on our squinting glasses at the stage of preliminary data, we can look at the deeper layers of the cortices, and we notice that in the seizure mice, and again, this is a representative example, in the seizure mice, we see possible dysplasias where it really does look like that in those deeper layers, there are bigger cells, higher densities of cells. And that's, that could be the sort of mousy correlate of the cortical dysplasia, which is thought to drive epilepsy in this particular disorder. So as conclusions, obviously we're at very early stages. I'm hoping that you'll be hearing a lot about this project in the next few years when I get at least one car attached to our tiny little engine. We have proof of concept that you can have a haploinsufficiency epilepsy model in mice. You don't need to engineer that second hit. And that's a huge deal because it allows us to do genetic mapping for this disorder. I don't know what to make yet other than it's like cool fact, this transgressive segregation of epilepsy. I was sure that we would see it in the DBAs. I guess maybe Rob knows, but it might be that the DBAs, it might be that all of those modifiers or many of them are recessive. I have no idea, but we certainly didn't see what I thought we would see. We do have heritable variation in TSC-associated brain abnormalities, and that could give us some sort of subthreshold endophenotypes to mess about with when we finally get to mapping. And so obviously the next goals are to map with various F1 crosses. I've got a few irons in the fire with respect to BXDs and diversity outbreds. Hopefully one of those will land, and I'll be back to talk about this project in the future. But we're really excited about the home cage behavior kinds of assays that Vivek Kumar talked about yesterday, because we do have this very stereotyped behavior presentation. We can notice when a mouse falls over, for example, and that's a pretty reliable marker of when it's having a seizure. So we can start to do things like monitor them for many, many days, many mice, multiple mice at a time, and count up seizures, track identities, and genetically map with those. Whereas like looking at hundreds or thousands of hours of video is not feasible, and you certainly, it's a real pain to do electrophysiology. Okay, so that's it. Thank you very much.